So it's the second part of the demonstration. I just explained that I've just shown in the first demonstration how to retrieve the data from papers that have been published in PubMed and whose chemical data have been uploaded into Campbell database using the direct interface from DataWarrior. How we label the compound to get the name of the companies, and how we use the built-in filters just to keep the compounds that have been tested on the CCR5 target and which have been tested with an IC50 test. Then we have extracted all the compounds from the paper published by Merck, and we displayed a 2D view that shows on the x-axis the structures and on the y-axis the results of the biological test, so the IC50 values, using a logarithmic scale. And what we are going to do today is to create an SRR, so a structure-activity relationship table or view, using the compounds of this file. And then we will compute the mean effect of the substituents found using box plots. And then we will sort the substituents according to their mean effect, so the effect they produce on average. And the goal is to obtain such a view. So a view where you display all the substituents on the X and Y axis, and where you can also, on top of this, sort the substituents according to their activity. So the activity here is displayed using the color, so the green marker are the good compounds, and the red marker display the less good compound, let's say. And this is also useful to identify the holes, what I name holes, so the area in your SRR where no compound has been synthesized, and maybe it would be interesting to do so because the other compounds, the similar compounds, show a good activity. And then we will also add some foreign compounds, so compounds from your dataset, for example. There is two methods for this. Either you add them manually, so one by one, by adding rows to the DataWarrior file, or you can also import them from an external file that you have prepared, so maybe your own results in a separate file, you can import them into this Campbell database file. Then we can also add additional compound properties, such as the CLOP. And once we do that, we have to reapply the SRR, so as to take into account these new compounds and new property. So now I'm going to go to the DataWarrior. Oops, no, it's hiding part. So first thing to do is to open the file we have created before. So it's this one, starting point. That is the point where we were left off last time. So you see the 2D view with the small compounds here and the value of the test. So what we would like to do, so this is, I have to hide this. So first thing, maybe we want to add some colors to this graph. And for adding color, you just right-click in the middle of the view and select Set Marker Color. Now we would like that the color green represents good compounds, and the color red, for example, would represent the not so good compound. So first we have to select the column which will be used to map the color. So in this case it's value, which contains the results of the test. And we have to change this because at the moment the smallest value have the red color. And the greatest value are blue, and I want to have them red. You can also change the way it gradiates by just clicking on the middle. And this is done. So if you go into the structure view, you see that the compounds here, they have all this core substructure in the middle that we want to use, and we would like to split all the compounds in 3 or 4 parts. So one part would be the core fragment, and all the other would be the substituents. So to do so, we have this nice functionality in DataWarrior, which is called core-based SI analysis. So I will just before copy one of the compound with right-clicking. I can do copy structure as a chemical structure. That was with right-clicking. Then I go again into this chemistry core-based SI analysis, and this is how the dialog looks like. So first you are required to select the column which contains your structure. In this case, there is only one column containing structure, so it's no problem. Then you have to define the core, and for this you have many different ways. If you right-click again into the middle of the pane, the white pane, you have 3 different options. The first one, Add, will open the auxiliary structure editor, where you can draw by hand your compound. But this is not what I want to do. I'm too lazy. So I will use the second one, paste. This is not the core, so I have to change it. Okay, that's also a little bit boring. I will do another method. Remove this one. Right-clicking, I can remove. I will just simply add it from a file that I have prepared. So it has extracted from the file all the core structure, the structure it has found in the file. In this case, there was only one. So now the core is defined, and I can go to the 2 next options. So I have the choice between distinguishing the stereoisomers or not, and to use the existing columns for the scaffold and the substituents. Since it's the first time we are creating the SRR, there is no existing column, so I can remove that. It would not harm if I would have left it. Distinguish stereoisomer is maybe useful, so I will keep it. So what happens is that it has created 4 new columns here at the end of the file, one containing the scaffold, one containing the first substituents, and so on, the 3 substituents from each one column. So if I go back to the 2D view, I can quickly see how many scaffolds and how many substituents there are by just changing what I'm going to display on the x-axis. So for example, scaffold, what kind of scaffold do I have? You see that there is this scaffold, which is the very well described stereoisomer, and the second one is the mixture of both enantiomers. Then I can go further. R1 contains all these different substituents, lot of different substituents. Every time there is only one. R2, there are only 2 variants of R2 substituents. Sorry. And R3 only consists into fluorine or nothing. So I will just increase the size of the window. Now, if you would like to know, on average, what is the effect, so the value here, of the fluorine addition or no addition of fluorine. It is very easily computed here using changing the chart type. So here, at the moment, we have a scatter plot. So every compound is represented by a square marker. But we can change the chart type by right-clicking and selecting, instead of a scatter plot, a box plot. And the box plot, okay, we have a huge point here. We can reduce it by, again, right-clicking, changing the marker size to a more reasonable size. So with this kind of plot, you immediately see the distribution of your data. The average is represented with the red line and the median by the black line. there is no numerical value of this median and average, so either you will display them by changing the statistical view option, or more easily you just copy these statistical values that are there in the plot but not visible. So now they are in the clipboard, and you go on Edit, Paste, and here they are. So you immediately see the two different kind of substituents. No fluorine, fluorine. 14 compounds had no fluorine, 15 had one. The mean value of no fluorine is 7. The one of the fluorine is almost 2. So you have all the numerical value directly in this new file. So it was just by right-clicking onto the header and selecting copy statistical value, then paste. If you just change the axis and go onto R2, same thing, you see that this substituent is not as favorable as this one, but this one has a very big variability. And for R1, it's not possible to display a meaningful box plot because every category has just 1 point. And the scaffold is the same. So if we want to display the same 2D view that I've shown you in the slide, we will be using on the x-axis R1, and on the y-axis, for example, R2. And now the marker size are too small, so I have to again adapt the marker size like this. And as you can see, they are not ordered by activity. This one is not that good, and the best one is in the middle of the axis. So we would like to have them ordered by their effect. So for this, I will again minimize the view, and I go on the table view. On the table view, you have this header of the columns where if you right-click on them, you have the possibility to change the category order. So it's not random or alphabetical as it could be. It's something that you can now change. You just have to click this Use Custom Order checkbox. Now you can manually order them by just drag and dropping them somewhere. Or you can also use this functionality to order them in an ascending order according to their value of activity. So here I've selected in ascending order by mean of the numbers which are contained in the column value. Then I have to click on Sort Category, and then the magic operates, and all the substituents are really ordered by their effect. Then if I click on OK, the 2D view is going to be updated. See, now on the x-axis, it's ordered by activity. And we can do the same for R2. Set category custom order, use custom order, again in ascending order by mean of the value column. I'm sorry, first sort. Then OK. So this is now the view I have shown you before on the slide. We have these empty spots, empty area that we could maybe try to fill if necessary. Maybe in this case it's a little bit too simple because these substituents have only one, have been only measured once. But in principle, you can use this for every SIR. So there is also the possibility to add other compound properties. It's in chemistry, so I was a little bit quick. In chemistry, there is the menu point Add Compound Properties, where you can add, for example, the lipophilicity of the compound. You just select C log P and click on OK, and a new column is created containing all these C log P. If you want to know more about this functionality, there is a help, and in the help there is this small underscore here which contains all the details of all the properties. For example, the distribution of the CLOP and how it was computed, which method, and also some papers who described what is the benchmark of this CLOP. So I won't click on OK again, otherwise it will create another column. Now if I want to add an additional compound, I have a compound I would like to include into my SRR. I will just copy something for me. So I will just go into data and then add an empty column. Oh no, not column, sorry. Data, add empty row, of course. Just one. Now you see we only have 29 visible compounds. On total we have now 30 rows. So one row is hidden. To show it, the best and easiest way is to disable all the filters. Then we are left with 30 compounds and we can scroll through the end of the file. And by double-clicking onto the structure column, I open the editor. I can paste the structure I've just copied before and create a new one. And maybe I have a value to add, and it's a very good compound, I'm sure. So that's how you can add some more compounds. And another possibility is to append the file. So it's an in file, append file, and then you select one file that contains also structures. And you have to map the content of this new file onto your existing file. So the new file contains 3 columns. One contains structures, the other contains IC50 own test values, and then there is a third column containing the comments. So with the first structure column, I will append this structure column to the structure one of my file. Sorry, it's a little bit. And IC50, I want to add it behind the value column. Because it's also IC50 value, I want to have them in the same column as the value column of my file. And then comment does not yet exist into this file, so I will just create a new column with it. Then I click on OK. And if you go through the end of the file, you see that this was the one that I've added by hand. These 4 compounds have been added by the appending of the file with the comment here and with the label telling that it comes from another file. And this is the label stating that it comes from the original file. So now if I want to place my new compounds onto the SRR table, at the moment they are all located here because they have no R1, no R2, it has not been yet split. I have to redo this core-based SRR analysis. So I select again the mystery core-based SRR analysis. Everything is already ready except that I will use the existing columns for scaffold and substituents, and I will overwrite and complete these 4 columns. And you see that now it did the job again to find the core and the substituents. Still, the 2D view is not updated. That's a bug in DataWarrior at the moment. To update the view, we have to save the file. And reopen it. And now it's okay. Sorry. Yeah. If we select these 5 compounds, we'll see where the selection is. So if now it's again not ordered by activity. You see that the green points are at the end. So we would have to repeat the operation of ordering R1 using the value that are in the column value. And then it's done. So if you want to see our compounds added from the external file, you just create a filter on the comment column, or even better would have been to set the filter on the dataset name, and then you can find easily where your compounds have been ordered into the SR view. So I think it's the end of the presentation. I was quick, but not quick enough. Yes. So, and last thing, as always, for questions, just remind that I'm not a chemist.
That's fantastic. Thank you very much, Isabelle. Does anybody have any questions for Isabelle? I'm trying to unmute you all so that you can ask them. Could get a bit noisy and chaotic. Any questions for... Oh, there are a couple of questions here that I've just spotted. Chris, do you want to ask your question? Okay, I can't hear Chris very well, so I'll ask the question. Chris has asked, is it possible to script DataWarrior, perhaps using Python?
Just wanted to say there is a macro mechanism inserted in DataWarrior where you can simply record what you are doing on the screen. And most of the tasks you are doing on the screen are supported by this macro mechanism, and then you can rerun the macro afterwards. But it's not with Python. It's really a standalone proprietary mechanism.
Okay, excellent, thank you. And a question from Eamon as well. Can you plot two properties, say activity and solubility, and then color code the data point by R group, say R1 red, R2 blue, etc.?
Yeah, I think it's not a problem. So you just change the x-axis and the y-axis, set that on C log P and the activity. And then you have to change the color and say that you want to use R2, for example. So now you have these two different colors for the two variations of R2. That's what you wanted to know?
I think so. Let me see if I can unmute and just...
Can I show again? So it's you select on x-axis and y-axis the two values you would like to display, and then you change the color according to the R1 or R2 or...
Yeah, that's great. Thank you.
So R1 is not a good choice. There are too many.
Okay.
And then, of course, you can also change the color if you want.
Excellent. Thank you. And Chris, did you have any? Did that answer your question about scripting?
It does mean that you can't access external programs then, I presume?
No. For the moment, it does not access external programs, and you cannot script them externally. I'm not sure if it is planned to have this possibility. I have to ask the developer, who is Thomas Sander. You can ask him.
Okay. If anybody has any other questions, please feel free to pop them in the questions panel. So
there is, sorry,
Yeah.
Just wanted to add that there will be in the future the possibility to add to use the NIME nodes for DataWarrior. So for the moment it's not yet open source, but this is a possibility. Yeah.
Can you just describe a little more about what that would mean?
You know, NIME, where you can describe a process by small tasks, and these tasks are named nodes. And we are in the process of making that open source. Some nodes have been written for DataWarrior, so they would be able to call DataWarrior, and so it would be a possibility to interact with DataWarrior from outside.
Excellent.
And to link the task you are doing with DataWarrior with other things.
Okay, that sounds very interesting. Thank you. That'd be excellent. And just as a little advert, we're talking about NIME in the next session of these in April, so it's very timely.
It's great.
Okay, fantastic. Thank you very much indeed, Isabelle. So I'm going to change over to Andy. I guess I should say also in just before handing over that there's a lot more functionality with DataWarrior, which you can see lots of examples of all kinds of interesting SAR analysis under the help menu option. If you look under there and start diving, you'll see a lot of interesting ways of doing SAR analysis, activity cliff analysis, and self-organizing maps and a whole lot of stuff. Anyway, let me now move over to Andy, and Andy is going to talk to us about the RSC, the work he's been doing with the RSC on Medicinal Chemistry Toolkit.
Okay, thank you for inviting me to come along and present this. So the presentation is from myself, and I worked very closely with Sula Armstrong at the Royal Society of Chemistry, and as you shall see later in the presentation, Alex Clark from Molecular Materials Informatics in Canada. So the idea for the Medchem Toolkit actually came from a conference I attended in Cyprus in 2011. Just prior to that, the RSC, the Royal Society of Chemistry, had asked me to edit the third edition of their book, Medicinal Chemistry Principles and Practice. And to tell the truth, I wasn't so interested in editing the third edition of any book. But at this conference, there were lots of talks from neglected tropical disease groups and not many people from industry. And at the end of the meeting, there was a round table, and it struck me in the discussion that what these groups wanted was access to industry-validated tools and predictive methods, pharmacokinetic, pharmacodynamic support, and maybe hints and tips. And so thinking about that book, I wondered whether we could make these tools available through an ebook format. And so I pitched to the RSC that we publish an ebook with embedded tools to help medicinal chemistry practice around the world. So the Handbook in Medicinal Chemistry and the Toolkit app was born. Now the ebook format is a little bit restrictive, and we felt it might be easier to develop the applications separately from the book itself. And actually, myself and the RSC were very happy that we could do this, because we both agreed we could make these tools freely available while RSC Publishing did their job of selling the book. So I don't really want to talk about the book. I'd love to say that it was freely available, but you have to buy it. But the app is completely self-sufficient and is out there on the iTunes Store for you to use. On a tablet, because it was meant to go alongside this ebook format. It was important to us, I think, that the apps in the tablet would run local calculations and didn't call out to external sources, so that it was safe from an IP perspective. So we had to decide on what platform we would present this, and I also attended the Dallas ACS shortly after this, and I was taken to a session by the RSC on mobile computing, and it appeared at that time that iOS was much the best place to develop a tool like this on a tablet. mainly because that iOS is a very consistent development environment. Apple controls everything. There's one operating system. iTunes allows you to push updates when they become available. And importantly, at that time, there were millions and hundreds of millions of iPads and iPhones available. Now, I guess now it seems that Android is rapidly overtaking Apple, and we kind of wished maybe that we had two platforms. But we don't. It's only available on Apple at the moment, but we'd love to port this to Android if we could. The other problem with a tablet-based system is if you wanted to put chemistry-enabled applications in, there was no toolkit for iOS, and that was a problem as we wanted to do some of these chemistry-enabled predictive methods. So this is what we wanted to deliver, some local calculations of physicochemical properties, log P, ligand, lick efficiency, and also include some of the things that AstraZeneca has published on ugly filters. So we wanted those to be available, and that needed this chemistry toolkit. We wanted to include some of the useful tools that I've used over many years with my colleagues at AstraZeneca, certainly the dose demand prediction method that I think rather revolutionized how we did drug discovery when we started using that method in the 2000s. And also some hints and tips, things I found useful in my time, in fact daily useful in the projects I work. So they're the things we wanted to do. And the toolkit's available. There's a number of methods here, and rather just walk you through it, I'd like to show you a couple of case studies from my own projects where using some of the applications has really helped us. So to start with, I'd like to just think about translational science, and so we should. It's a very important aspect of what we do. So many of our projects fail so late in development, largely because of the failure of our mechanistic hypothesis. So I guess what we're trying to do is trying to find targets linked to disease, identify a potential mechanism underlying the disease, and evidence that that mechanism is indeed driving disease, and then that can be a target on which we work. So then we develop an assay screening cascade where we can identify and optimize compounds in really simple assays, show those compounds work in cell assays, and even in maybe more complex cell assays representing diseased tissue. Show that we can control PK/PD, and then take it to the final experiment where we possibly only test one compound in an N of 1 experiment that may cost us $125 million. So as chemists, we optimize compounds on the top assay in the hope that they will predict efficacy in the bottom assay. So translation of activity is really important. And the problem is that we often see problems in that translation, seeing that the assay we use to optimize our compounds understand how the activity changes as we go down that cascade. You may see differences between assays, and the reasons why can take the project in many different directions. It may be that there's just some differences in the way the assays are constructed, and we must be careful not to be misled by these. We should ask how our assays are configured in a lot of detail. It may be that the permeability or the physical properties of our compound is wrong, in which case that's what we should optimize. Or it may be that the intrinsic state of the protein or target in our target cell is different from in our screening assay, in which case we want to make sure we screen our compounds in very relevant assays. So here's a very typical problem, and it is from one of my projects, where we have an enzyme assay and a cell assay that correlate, but there's a big drop-off between the enzyme potency and the cell potency. And so the question is, which potency is the real one? Which one should we use to predict the human dose? Can we understand the differences between these assays? So this is a rather straightforward one. It's a P38 kinase problem. And we know that our compounds are competitive with ATP at P38. So as we change the ATP concentration, the observed activity of our compounds will change. So what's the IC50 in our enzyme assay? What's the IC50 in our cell assay? And can we understand the difference? So we can think about this in terms of a simple substrate competition way. And the Chen-Prosov equation kind of describes that mathematically. So one of the first apps we wrote was one that enables us to understand differences in activity in substrate competition experiments. So in this case, for P38 in these compounds, our assay was run at 100 micromolar ATP. We know the Km for ATP at P38 is 25 micromolar. So putting in the activity of our compound in our enzyme assay, we can then predict what the activity would be in our cell assay, where the ATP concentration is around 3 millimolar or something. And we predict the 24-fold shift in potency on going from our enzyme assay to our cell assay. So we can see in this P38 example, if we allow for a 24-fold shift, that's the drop-off we expect on going for our cell, then there's a very good correlation between our enzyme assay and our cell assay in this case. And that tells us a lot of things. It tells us the enzyme is very good at predicting the cell. It tells us there's no permeability problems in terms of this cell assay with these compounds, no efflux, no uptake. And then we hope that will also translate into a target cell assay, which of course we went on to follow. So that's the first step. Here's another example that's very similar in this case study. It's another one of my projects. It's not the same one as previously. We have an enzyme assay and we have a cell assay where there's a drop-off between the two. So can we understand the drop-off? Is it understandable? Which activity should we use to make our human dose predictions? So in this case, there's a somewhat different reason. In this case, when we asked the biologists how they constructed their cell assay, they had added a little bit of human serum albumin to the assay, not very much, 0.1%. And so maybe the protein binding of these compounds causes that degree of shift between these two assays. And this is a really common finding. Cell assays often have small amounts, things like fetal-calf serum or human serum albumin or bovine serum albumin in it to keep the cells happy. And these proteins non-specifically bind most of our drug molecules to a greater or lesser extent. And so we need to know about this because in our dose predictions, it's the unbound potency that we need to use to make our human dose predictions. So we should allow for the fact that there might be a small amount of binding in our cell assay. So one of the second apps we put in there was to calculate the potency shift due to plasma protein binding between an isolated protein assay and a cell assay, and even if you go to whole blood. So in this case, we know that plasma is around 4% weight per volume human serum albumin. And in our cell assay, we had 0.1% human serum albumin, which is about a 40-fold dilution. So of course, every compound will shift depending upon its protein binding. So I've chosen one of the compounds here. Its free fraction in plasma is 0.0061, so it's 99.4% protein bound. And the cell potency we measured was 2.4 nanomolar. So if we put that into this little app, it will tell us that its free cell potency is 0.47 nanomolar. And in fact, you can calculate what the whole blood potency would be. So if we did that for every compound on that previous graph, and we see how the correlation changes, then I can show you on the next graph. So now the PBMC potency is much more aligned to the enzyme potency. So actually, the line doesn't go completely through the center of all the data, so it's not completely described the difference between the enzyme and PBMC potency, but largely it has. This is real data, so it's not made up. This is what we found. So we were able to scale our human dose prediction off of our unbound cell potency, and one of the compounds on this plot we've just recently taken into the clinic. So the third part of the app I'd like to show you is probably the one that can change most the way I thought about drug discovery, and this was the dose-to-man app that we developed at Charnwood in the between 2000 and 2010. Now, when we optimize drugs, we're actually trying to do a multi-objective optimization. You're trying to keep the minimum plasma concentration of your drug above the minimum level, which we could call the minimum effective concentration, which would be some multiple of your cell potency, your free cell potency. We want to do that with once or twice a day dosing frequency probably, and to maintain a good margin between the concentrations you achieve in plasma and some side effect level. So my colleagues at Charnwood started to use this mathematical analysis of dose to man, and using this, I'll say, simple equation. It's a little bit complicated, but I'll talk you through it in a moment. they studied 63 drugs on the market and found that from a few simple measurements, they were able to predict the clinical dose, the clinically used dose of these 63 drugs within an acceptable error margin. And in fact, most drugs on the market, their minimum effective concentration is in between 1 and 3-fold the cell activity they found. So we coded up this dose-demand prediction method within the app, and we've made it available for you. So I show you here with fexofenadine as one of the examples, and in fact it is one of the examples in the book, which is where the data came from. So actually, from only 5 measurements that you can make within a couple of weeks of making your compound, you can make a human dose prediction. So here for fexofenadine, you'd need primary potency. You need an estimate of human clearance, which you will get from a human hepatocyte intrinsic clearance assay. You need an estimate of the human volume, which will come from the volume distribution from a preclinical species, such as rat, and knowledge of the difference in protein binding between rat and human. So they're the sort of 5 measurements you need, and then some assumption of how much you want to cover your cell activity as a minimum effective concentration. So for fexofenadine, as we felt between 1 and 3-fold the IC50 at Cmin was where we'd like to look. So that will give you a predicted dose using our app, putting the numbers in, of between 0.7 and 2.3 mg per kg per day. And the actual dose used for fexofenadine is around 1.7 mg per kg per day, so right between the predictions made by the app. So you see, but just using a few simple measurements you can make, you think of a compound, you make it, within 3 weeks you can be making quite accurate human dose predictions. And I can now show you the app and show you how we can illustrate a common misconception around drug discovery and drug optimization. What happens if we increase the protein binding of fexofenadine? So I'm going to change the human protein binding of fexofenadine by 20-fold. I'm going to increase it by 20-fold. And we're going to see the effect it makes on our human dose prediction. So I'm going to change the protein binding of fexofenadine by 20-fold. So taking it from 60 to something like 98%. And just watch what happens with the rest of the numbers. Okay, it has changed. So it's now at 98%, and the estimated human volume has changed, the estimated human clearance has changed, the minimum effective concentration has changed, but actually the half-life and the dose are almost constant. This shows you that in an open system in vivo, changing protein binding has very little effect in most cases on the human dose and half-life predictions. That's not what happens in vitro, of course, because you'll see a shift, you change protein binding. In vivo, it has almost no effect. So now we take our life into our hands. That's the dose-demand calculator. Every part of the app has a quite comprehensive help text associated with it. So here's the one around the dose-demand calculator. And in fact, the whole app itself goes through and takes you through the drug discovery process when you might find certain apps useful to you. So the final part of the Medchem toolkit was this chemically enabled functionality. And now I'm going to try and run a video here, Mark, so we'll see what happens. It's running through. So this was the most difficult part of putting this together because there's no chemistry toolkit available for iOS. So we looked at a number of chemical structure drawers that were available on tablets, and one of them was developed by Alex Clark, and he developed that for ChemSpider, which is the RSC's searchable database. And actually it's a really nice, slick chemical structure drawer. So we used this basis for our chemical property calculator. Alex is in Canada, we overlapped late at night, and it was many, many months of work that ended up producing this. It's a lovely little tool. As you draw, it calculates physical properties for you, and you can see the AstraZeneca ugly filters. I'm just going to run it again as we talk through. It may not go back. Yes, it's going to go back. They update in real time. telling you, based on the years of experience of many chemists at AstraZeneca, things they like to see or do not like to see coming out of high throughput screens. So at the moment it says filter 1.6, no polar atoms. It doesn't like to see really bland structures. And then as we draw the structure, it continues to update and ends up at the end. We get there in a moment. As you see, we can put in the potency right at the end, and it gives us the ligand efficiency and ligand lipophilic efficiency. It ends up, and the only thing it doesn't like about it is the alkyl thiol. So that was really nice collaboration between us and Alex. So the app is out there, it's on iTunes, we'd love you all to use it. Allows you to control physical properties, understand assay translation, focus on dose demand predictions, and that was really important for us, and working really closely with the NPK at Astra Charlotte. This app was I was helped by many, many colleagues at AstraZeneca and outside AstraZeneca, and they're listed there. And just on the right-hand side, I got it up Paul's paper on nutrition. And if you want to play drug discovery companies, you can see what happens when you change cycle time, number of projects in different phases, and costs to develop them. So thank you, and I'd like to hand it back to you, Mark. And if there are any questions, I'll be happy to answer them.
That's fantastic. Thank you very much, Andy. Just while we wait for a couple of questions to come in, I'll also launch that question that we talked about, you know, obviously relating to the platform that you mentioned in terms of iOS, and really just checking out for what proportion of people that would really, at least of this audience, would that prevent you using it. So let me see if I can unmute Chris, who has a question, while we just do that. Chris Swain, you want to ask your question?
Well, is there any way to add further Medchem filters?
Yes, in principle there is. In practice, so I developed it with the RSC. Clearly, this cost quite a bit of money to put together. It started off with myself coding up as an amateur, but we have professional help to make it robust and suitable to put on iTunes. It's really the cost of adding new things to it now, and whether there's some way we can do that. But of course, technically it's possible. We'd like to add other filters into it, Payne's filters, for instance, and there are other useful apps we'd like to add in as well.
Okay, thank you. Charlie, you have a question. Oh, looks like you might be muted, Charlie, whether you want to ask your question. If not, I can do it. Charlie's question is the toolkit only available on the UK App Store? I can't see it in Switzerland.
No, it should be available on the World App Store, actually. So it should be available in Switzerland. If that's a problem, I'll contact the RSC. You'd probably have to search for it on an iPad. If you looked for it on an iPhone, it wouldn't show because it's an iPad-only program at the moment.
Okay. Charlie says he'll try again with an iPad.
Yes. It'd be great to have some of the apps at least available on an iPhone.
Let me just share that poll. So I guess at least of this audience, you know, just over half would have access to iOS, but you know, a significant proportion, I guess, would be a problem, including me, until I buy an iPad. Okay, any other questions from anybody? All right, that's fantastic. Thank you again very much to Andy and to Isabelle. And next month's presentation will focus on NIME, and we do have somebody from NIME giving the introductory presentation, and then a couple of people talking about applications. So hopefully you'll be able to join us then. you'll get the usual follow-up and link to the video, and hope to speak to you again in a month. Oh, I have one more question. Sorry, Andy. Is there any plan to move it to the Windows platform as well?
It's the same answer really about cost of doing it. It would be lovely to make it available on all platforms, and I think if there will be a point where the RSC would feel it's worth doing that, and that'll be based on the number of users on the iPad platform. You can see how we came about to start this, that it was meant to go with an ebook, and therefore it was a tablet that we chose as a way of delivering it. But of course it could be available on all platforms, PC, Android and iOS. It would be fantastic if that was the case, but at the moment, no.
Do you think there's anything that this group or anyone in this group could do to help?
Yeah, it's really about funding. If we could work together with the Royal Society of Chemistry to make this available on multiple platforms, then I think they'd be very willing to consider doing that. The desire is there. They would love to work with me to make this available on Android as a start point. But it's really the cost, not just the cost of doing it, but the cost of maintaining it as well.
Yeah. Okay. I think one question I might add into the follow-up email, which people will get tomorrow, would be, you know, would you rather see it on Android or Windows? Would that be a reasonable question? Just to, you know, if there was money to do one but not the other, what would people's preference be? It's a limited and biased sample, but anyway.
Yes.
Okay. Thank you very much indeed, everybody. That's been a really interesting session, and thank you once again, particularly to Isabelle and Andy.